Hi, welcome to Animated Biology of the Cell. Today we discuss DNA double-strand breaks, the mechanisms by which they are fixed, and the consequences of these different methods. I knew from the start that this was going to be its own video, partly due to the fact that there are 4 repair mechanisms to be described, and partly because these mechanisms are sometimes difficult to wrap your head around. Central to this video is going to be the following graphical representation of under which condition each mechanism gets used. I will start with non-homologous end joining and then go into end resection for a little bit and then use that as a jumping off point to talk about alternative end joining, single-strand annealing, and then talk about the most complicated one, homologous recombination. So, a double-strand break occurs. What does the cell do? A brute force solution to fix a strand that is snapped open would be to stick it back together again. And this is exactly what happens most of the time. It is called blunt end ligation. 2 Ku proteins find both ends, bring them close, and ligase 4 pastes them back together. It is quick, requires very few proteins, and seems foolproof, right? Yeah, that's all true, but what about breaks that don't have blunt ends? Blunt end ligation is only part of what's called non-homologous end joining. Other pathways are needed when the DNA ends are damaged by the break somehow. From the moment a strand break occurs, and Ku attaches to the broken ends, a ton of things can happen depending on the shape of those ends. Overlaps that contain incompatible ends for ligation will have to be trimmed, and then the resulting gaps in the annealed strand can be sealed by polymerase lambda and ligase. And if I may interject in between these explanations, during the course of this video's research, I realized that for a lot of these scenes, I was going to have to animate tons of proteins interacting with one another and communicating with one another, and I really didn't want to do that. Partly because I was not going to be able to animate all of them, and partly because it's not exactly necessary for the explanation of how these mechanisms work. I want you to understand the DNA and how the DNA reacts to this situation, what kind of fingerprints are left. Proteins are secondary. If you want to get into the nitty-gritty of which protein does what, there are sources in the description. In cases where the 2 ends are completely incompatible with one another due to significant decay, polymerase mu will actually put nucleotides on the strand in order to make them stick it back together again. This, of course, creates insertions, but apparently it's better than having your DNA split. So, when we compare the before and after pictures, you might notice that in quite a few cases, non-homologous end joining introduces mutations, especially when considering scenarios that aren't blunt ends. Despite this, non-homologous end joining is the most frequently used method of dealing with strand breaks. Why this is will be revealed when we talk about the next 3 methods. Let's talk about the cell cycle. For our purposes, the cell cycle consists of 4 distinct phases, all with their own duration. There's G1, when the newly divided cell has 1 copy of every chromosome and is focusing on performing its duties. In S phase, preparations are being made for the upcoming division. In this case, that means that all chromosomes are bunched together and are duplicated. And after this, in G2, the cell is relatively stable again, preparing for M phase, where division happens. So there are 2 relatively stable phases where we can find identical chromosome copies. This fact hasn't escaped evolution, which is why it will use this copy as a template to end up with a flawless repair. This process is homologous recombination, and we'll get to the entire process at the end. Just know that at the start of homologous recombination, large chunks of DNA on both sides of the break will be removed. This removal is called end resection, and it happens primarily in S and G2 phase, and that lines up with the fact that homologous recombination happens primarily in S and G2 phase. It is unscientific to say that this process is only meant for homologous recombination, but one gets the feeling that it is that way, because the next 2 mechanisms that I'm going to discuss need end resection to function, but they have far worse outcomes when you compare them to both homologous recombination and non-homologous end joining. Let's talk pathways. From the moment a double-strand break occurs, non-homologous end joining is the default option. Even in S and G2 phase, 80% of all double-strand breaks are handled by non-homologous end joining. From then on, it really depends on whether or not homologous recombination is available. A lot of the mechanisms here rely on competition to see which one gets chosen, and homologous recombination just has the most abundant proteins. But if they're not available, homologous recombination cannot happen, it is deficient, and so either alternative end joining or single-strand annealing occurs. Alternative end joining relies on patches of roughly 4 to 10 bases on both sides that have the potential to pair with each other. By what mechanism is currently unknown, the loose ends can be trimmed before polymerase theta fills the gaps to the left and to the right. Single-strand annealing, while quite similar to alternative end joining, occurs mostly in patches of DNA which are highly repetitive. It will use these longer repeats to anneal the two strands together. How the two ends find each other is, again, not known exactly. After annealing, the overhanging bits are cleaved by XPF and ERCC1, and any gaps are sealed by polymerase. Not polymerase theta in this case. Now, it might be important to highlight the differences between these two, because on the outset, they look quite similar. So the key difference that sets these two apart are the proteins involved. RAD52 seems essential for single-strand annealment, and might be connected to how the bases connect. But it is kind of useless for alternative end joining. Polymerase theta is an integral part of alternative end joining, but it does nothing in single-strand annealment. In experiments where RAD52 was removed from a cellular environment, alternative end joining activity went up and single-strand annealment went down, and vice versa with polymerase theta. Now, a second difference between single-strand annealment and alternative end joining is the damage that these pathways do to the DNA, the so-called fingerprints left behind. Single-strand annealment pulls two strands together and cuts off the overhanging bits. As such, massive deletions are quite common. Up to 25,000 base pairs have been recorded to have been removed. The trade-off is that this happens on highly repetitive regions of DNA, where these kinds of deletions aren't terribly hurtful because they aren't expressing many genes anyway. Now, alternative end joining, on the other hand, leaves a lot less deletions, but because of the schizophrenic behavior of polymerase theta, which can rapidly switch between template-dependent and template-independent nucleotide addition, alternate end joining can leave insertions as well as deletions. As you might imagine, these pathways can be even worse than non-homologous end joining in how mutagenic they are. This, combined with the fact that the rate of occurrence rises when homologous recombination is out of the picture, makes scientists believe that these are backup systems to homologous recombination. Homologous recombination. It starts with end resection. The single strands, literally by trial and error, find the exact spot on the sister chromatid where it can use the sister chromatid as a template to get back the nucleotides that it lost. The structure that is formed here is called a D-loop. Now, D-loops are important because their stability determines the outcome of homologous recombination. An unstable D-loop will collapse before the second end can anneal to the other loose hanging strand of the sister chromatid. This way, after collapse, the correctly extended strand will anneal to the right place and any gaps can be sealed by ligase. This is the perfect repair that we are looking for. I mean, look at that. It's a perfect loop. This is what we want. There are even proteins working in the background that make this D-loop as unstable as it needs to be. A very stable D-loop, however, can also be desirable in different circumstances. In the case of a stable D-loop, the other single strand will then also anneal to the sister chromatid. A polymerase will then also go to this end, extend it a bit, and then leave. This is regrettably not called a double D-loop. It is a double Holiday junction, named after Robin Holiday, who described these junctions. And at this point, I have shown you in DNA form how the situation is established, but I think that for clarity and also for my own animation skills, it is easier to go more abstract. By tracing the DNA with green and yellow lines, we can keep track of the individual strands, but more easily observe the consequences of the actions that can be taken to resolve these Holiday junctions. Now, these traces almost match with what you would see in a textbook when they show representations of double holiday junctions. I just need to flip the DNA in a few places so to accurately match these drawings. So now we'll leave the nucleotides behind and go full abstract. The way cells deal with double holiday junctions is by placing cuts at specific locations so that the two chromatids become disconnected. If we move the camera to a specific place and cut out the second junction for now, I hope that it is obvious that the model here is the same as the abstract model. To keep the strands intact, cuts in this model should only be made diagonally, like this and like this. Going back to our abstract model, that means that only these two strands or these two strands can be cut. These cuts can be made independently of one another, so there are 4 ways to cut. The 2 symmetrical cuts are the least convoluted. The strands that are cut simply go over to the other side. These strands are usually identical, so nothing seems to have happened. This is good. An asymmetrical cut will do a lot more. Because of the way that the strands are still connected, here and here, it will actually flip the strands on one side, resulting in 2 chromatids that have exchanged strands. This is called crossover. During sexual reproduction, crossover is more important, as it can determine the traits of offspring. But that is a topic for another video. And with that, we have reached the end. Thank you so much for watching. I put a ton of effort into this one to improve my style. Please, let me know in the comments, what do you think? And how this video kind of felt like in contrast to my other videos. See you next time.